Okay, for this week, we will be covering the three chapters that will include the introduction to oxygen and carbon dioxide physiology. We will talk about the external respiration, that's the physiology of breathing, and we will be talking about the transport of oxygen and carbon dioxide in body fluids. So with an introduction to some of the acid-base physiology. So starting with the introduction to oxygen and this carbon dioxide physiology. So you can see here that there are the two kinds of organisms. You can see the coral reefs and the beetle in this figure here. So these aquatic animals are collecting the oxygen from the water in which they live. So the oxygen is actually dissolved in the environmental water, and it must move from water to each animal cell for the cells to use it. So both of these kinds of aquatic animals, they actually take up the dissolved oxygen from the water by breathing with the gills. So you can see the orange pine tree shaped structures. These are the gills of coral reef worms. So these worms, often called the Christmas tree worms, live in tubes that are embedded in the corals. So they might need some oxygen by projecting these gills, so consisting of highly vascularized tentacles into the water. So what happens that these tentacles, they also collect some food. So here in this figure here, you can see the beetle. So these diving beetles are common. So the diving beetles, which are common inhabitants of freshwater ponds, you can see here the beetles. So they are actually the common inhabitants of these freshwater ponds. So ostensibly, they breathe by acquiring the bubbles of air from the atmosphere. So, however, we can see a beetle's bubble also serves as a gill to acquire oxygen from water. So, while the beetle is underwater, and this gill action then accounts for most of the beetle's oxygen uptake during a dive. So these aquatic animals are actually collecting this oxygen from water in which they live. So we are talking about the coral reef worms and the beetles. So these are the coral reef worms and this is the beetle. So the oxygen is dissolved in the environmental water and it must move from water to each animal cell for the cells to use it. So we have already discussed the movement of gases. So we will follow the same convection uh, we adopted during the chapter 5 that was about the movement of the gases, and we can use the word transport as a general term to refer to all the movements. So thus, for oxygen to move from environmental water to the cells of an animal, conditions must be favorable for passive transport towards the cells doing the entire path that oxygen follows to reach the cells. So coral reef worms. So talking about the coral reef worms in the photographs we have, which have the gills that are composed of these spectacular pine tree shaped arrays you can see. So these are the arrays of the tentacles. They have an elaborate circulatory system. That circulatory system is actually filled with blood that is rich in hemoglobin like oxygen transport pigment. So as their blood flows through their gill tentacles, so these tentacles, they act as the gills. So oxygen enters the blood from the environmental water by passive diffusion, provided conditions are favorable for passive transport in that direction. So then the circulation of the blood delivers the oxygen to all cells. This is how the oxygen moves from the water by breathing wheel gills to the different cells. So one more point I want to add here is that the respiratory gases that we have, like oxygen and carbon dioxide, they are called the respiratory gases. So these respiratory gases move from place to place principally by two mechanisms. One is the simple diffusion, and another is the convection, or we can say the bulk flow. So in these cases, in the case of oxygen, these in fact are the only mechanism of transport, because as already stressed, active transport of oxygen is unknown. So carbon dioxide, however, is sometimes actively transported across the cell membranes. It might be in the form of bicarbonates formed by the reaction with water. We all know that when carbon dioxide reacts with water, it forms bicarbonates. So this movement, this active transport of this carbon dioxide in the form of bicarbonates takes place. So talking about the properties of gases in gas mixtures and aqueous solutions. So let's start with the properties of gas and gas mixtures. So modern study of gases in the gas phase traces back to John Dalton. That was a scientist who articulated the law of partial pressures. So according to this concept, the total pressure that's exerted by a mixture of gases, such as the atmosphere, is equal to the sum of individual partial pressures, or we can say individual pressures that are exerted by each of the several component of gases in the mixture. You can see here in the figure, for example, we see here the what is 1 atmospheric pressure that will be equal to the partial pressure that is exerted by oxygen, nitrogen, argon, or carbon dioxide. So it's like 20.95 by oxygen, and nitrogen comprises the partial pressure of 0.7808 atmospheres, and then we are talking about argon gas, that's about 0.0093, and the carbon dioxide is 0.0004. So when we sum up all these partial pressures, the individual gases like oxygen, nitrogen, argon, and carbon dioxide, we sum all these, and sum of all these will be equal to 1 atmospheric pressure. So the diagram here shows a container surrounding a body of dry atmospheric air at sea level. So data on the four most abundant constituents of dry air are shown. So the alternate exerts, you can see the exerts a total pressure of like 1 atmosphere. So which in turn, I already explained, that is the sum of all these partial pressures of the individual gases. So each constituent would actually exert its same partial pressure, even if the other constituents are absent. So an important property of partial pressure of each gas in a mixture is that it is independent of the other gases that are present. So moreover, in a volume of like mixed gases, so we are here talking about the gas in gas phases. So moreover, in a volume of mixed gases, each component gas behaves in terms of its partial pressures, as if it's alone occupied by the entire volume. So we can say the partial pressure of each gas in a gas mixture can be calculated from the universal gas law. That is, there is a specific reaction for that. That means PV is equal to nRT. So where P represents the pressure, V is the volume, N is the number of moles of gas, R is the universal gas constant. So there is a specific value for that, and T is the absolute temperature. So we can use this universal gas law to calculate the partial pressure of a particular gas in a gas mixture. So moving on to the gases in aqueous solution. So the partial pressure of a gas dissolved in an aqueous solution or in any other sort of solution is defined to be equal to the partial pressure of that gas in a gas phase with which the solution is at equilibrium. So to explain this, let's consider what happens if oxygen-free water is brought into contact with air containing oxygen at a partial pressure of like 0.21 atmospheres. So let's assume that specifically the volume of air is so great that as oxygen dissolves in the water, there is no change in oxygen concentration of air. And thus, the partial pressure of oxygen in the air remains the same, like 0.21 atmospheres. So oxygen will dissolve in the water until equilibrium is reached with air. Then the partial pressure of oxygen in the aqueous solution will be 0.21 atmospheres. But if this solution is later exposed to air that contains oxygen at a partial pressure of 0.19 atmospheres, the solution will lose the oxygen into the air until a new equilibrium is established. So we can say the partial pressure of oxygen in this solution will then be 0.19 atmospheres. So here we can use the term tension sometimes, which is used for partial pressure when speaking of gases in the aqueous solutions. So you can see with this in mind, we can say that three important characteristics of gases dissolved in the aqueous solutions come to light when absorption coefficients are examined. So number one is the solubility of different gases are different. So carbon dioxide has a far higher solubility than oxygen, or we can say than nitrogen. So the second point is that solubilities of gases in aqueous solutions decrease strongly with increasing water temperature. So this is true of all gases. So we can take an example like in this figure you can see to illustrate the absorption coefficients of oxygen in distilled water like at 0 degrees centigrade, or we can say 20 degrees centigrade and 40 degrees centigrade. The absorption coefficients of oxygen are 2.2, 1.4, and 1.0 millimoles per liter respectively. So we can say that if the partial pressure of oxygen is 1 atmosphere, a liter of distilled water dissolves 2.2 millimoles of oxygen. So when at 0 degrees centigrade, but less than half as much as 1.0 millimoles when at 40 degrees centigrade. So gases actually tend to come out of solution and form bubbles as water warms. So you can see here a sealed sample bottle of Cold Creek water after sitting in a warm room. So the bubbles form when water warms because a rise in temperature drives the dissolved gases out of the solution. And in case of figure B, you can see, let me explain this to you how it happens. So each grain of the cell increases the salinity in its vicinity, so driving the dissolved carbon dioxide out of the solution. So let's explain that how it happens. So solubilities of gases in aqueous solution decreases with increases salinity. So that means when we add these grains of salt, it increases the salinity, driving these dissolved carbon dioxide out of solution. For example, the absorption coefficients of oxygen at a fixed temperature in distilled water is 80% millimoles per liter. And we have full strength seawater that is 2.2, 1.8, and 1.7 millimoles per liter respectively. So increasing the salinity of an aqueous solution actually tends to drive gases out of solution by decreasing the solubilities of the gases. So this is a phenomenon called as salting out effect. So moving on to the diffusion of gases, we will talk about the function of a water beetle bubble as a gill oxygen, as a gill. For example, oxygen levels in the atmosphere, water and bubble gas, and oxygen diffusion. So simple diffusion, as earlier noted, is one of the two principal mechanisms of respiratory gas transport. So when gases diffuse from place to place, they do so by the same fundamental mechanism by which solutes diffuse through solutions. So the fundamental law of gas diffusion is that we all know that gases diffuse in net fashion from areas of relatively high partial pressure to the areas of relatively low partial pressures. So this is true within the gas mixtures. So within aqueous solutions and across the gas-water interfaces. So that means the gas is always diffuse in net fashion from the areas of relatively high partial pressures to areas of relatively low partial pressures. And we know that this is true in case of all gas mixtures and within the aqueous solutions and across the gas-water interfaces. So in view of this law, the coral reef worms we discussed earlier at the start of this chapter, they can get oxygen from their environmental water only if the partial pressure of oxygen dissolved in the water is higher than the partial pressure of oxygen dissolved in their blood. As we all know that blood flows through their gill tentacles. So similarly, the water beetle, like here in this picture, you can see in this figure, you can see that the water beetle that can get oxygen from the water only if the partial pressure of oxygen dissolved in the water exceeds the partial pressure of oxygen in its bubble gas. So let's consider an example of beetle. So here you can see we have to the dark portion here represents the partial pressure, and the light portion, the light blue portion here represents the concentration. So this is the partial pressure and concentration of the atmosphere, and this is the partial pressure and concentration of bubble gas, and this is the partial pressure and concentration of water. So let's consider a beetle that grabbed a fresh bubble from the atmosphere a while ago and has removed about 50% of the oxygen from its bubble. You can see here in this figure, the partial pressure of oxygen in this beetle's bubble gas will be about zero point 0.1 atmospheres. Like here, you can see the partial pressure will be zero point 0.1 atmospheres. The beetle bubble, in other words, will act as a gill. So this is true even though the concentration of oxygen in the beetle's bubble gas is like 4.3 millimoles per liter, which is far higher than the concentration in the water. That's 0.3 millimoles per liter. So oxygen will travel from high partial pressure to low partial pressure as it always does. But in this situation, involving both gas and liquid phases, it will move from low concentration to high concentration. So always equilibrium is attained with respect to any gas given when the partial pressure of the gas is uniform everywhere in a system. So like I already said, in this figure, the upper portion, the upper here, the dark portion, it represents the partial pressures, and the light blue is the concentration. So beetle acquires each bubble from the atmosphere. The beetle is assumed to have used about 50% of like oxygen originally in its bubble. So volume of the bubble is assumed to stay constant as the oxygen is withdrawn. So this is approximately true because the principal gas in the bubble, because of the principal gas in the bubble. So nitrogen tends to remain in the bubble even as oxygen is used. Temperature is 20 degrees centigrade everywhere. So we have so let's move to the other slide, which explains the principle of gas diffusion that are vital knowledge for the scuba divers. So what actually happens is that the partial pressure also governs the diffusion of nitrogen, a topical crucial for understanding the threat of decompression sickness in scuba divers and dolphins. So when a scuba diver, so we are talking here about, we are taking an example of a scuba diver. So when a scuba diver is at any particular depth, his or her lungs are kept normally expanded by the operation of the scuba air regulator, which ensures that the air in the lungs is at a pressure equal to the ambient, or we can say environmental water pressure at that depth. So Suppose a diver is at a depth of like 20 meters, so where the ambient water pressure is 3 atmospheres. So total air pressure in the diver's lungs is then also 3 atmosphere, and the partial pressure of nitrogen in the lung air is 78% of the total, or we can say 2.3 atmospheres. So if the diver remains at 20 meters long enough for his or her blood to come to equilibrium with the lung air, the partial pressure of nitrogen dissolved in the diver's blood will be 2.3 atmospheres. So this is not a problem as long as the diver stays at depth. However, the high nitrogen partial pressure will force the macroscopic bubbles to form in the diver's blood. So if he suddenly comes to the surface, so the reason behind that can be seen by imagining that an extremely minute microscopic bubbles they form in the blood. So that might be the reason behind that. So how do gases diffuse far more readily through gas phases than through the aqueous solutions? So what's the reason behind that? So the ease with which the gases diffuse is far greater when they are diffusing through air than through water. So there was a Krog diffusion coefficient for oxygen, for example, it's about 200,000 times higher in air than in water at 20 degrees centigrade. So one way to see the enormous implication of this difference is to consider, or we can say that oxygen that is diffusing between two regions that have a particular difference in oxygen partial pressure. So if the diffusion is occurring through air than water, the length of diffusion path can be 200,000 times longer and still have the same rate of oxygen transport. So that might be the reason for this difference. So an interesting application of these principles is provided by analyzing the oxygen supply to mice in underground burrows, or the oxygen supply to the eggs of sea turtles. Let's take an example of these underground burrows, mice that are in underground burrows, or we can take an example of the oxygen supply to the eggs of sea turtles. So what happens here is that oxygen, so we can say that if the soil or the sand is dry and porous, the oxygen is often supplied by diffusion directly through the soil or sand. So more specifically, oxygen is supplied by diffusion through the network of minute gas-filled spaces among the soil or sand particles. So there are actually the countless different paths, or we can say the countless angles of approach by which diffusion can occur from the atmosphere to the underground animals or eggs through the soil or sand. So what happens is that when transport along all of these paths is summoned or added, the diffusion through the gas-filled spaces in soil or sand often proves adequate to meet these oxygen demands. So this is true, however, only if the spaces among the soil or sand particles are gas-filled. So if the spaces become water-filled, that means there is some waterlogged condition in the soil or sand. The rate of oxygen diffusion drops by a factor of about 200,000. So a circumstance that can have drastic consequences for the underground animals or eggs. You can see here in this figure. So this figure here shows you the female loggerhead sea turtles that come ashore to lay their eggs on ocean beaches. So they actually bury their eggs about 40 centimeter deep in the sand, cover them and leave. So the eggs are dependent on oxygen diffusion through the sand to obtain oxygen during the long period of like 50 to 70 days. They acquire to become mature enough to hatch. So the nest described here, which contained like 117 eggs, was positioned unusually close to the surf's edge, and the sand overlaying the nest became submerged underwater when the eggs were about 54 days old. So the oxygen posture pressure in the nest was measured at regular time intervals with the fiber optic probe permanently implanted in the nest. So at the time of hatching, most eggs were dead when hatching occurred 10 days later. That means because the oxygen they didn't get enough oxygen. So what happened that for the first 50 days when the sand was mostly dry, the oxygen posture pressure in the nest was high. You can see here the y-axis, it represents the posture pressure of oxygen. So for the first 50 days, the posture pressure was good. So it was increasing. So they had good oxygen concentration. But the oxygen posture pressure fell to 0 for 2 days when the sand became temporarily saturated with water. That means the spaces between the uh, soil particles or sand particles that was saturated with water. So there was not enough gas exchange through those pores. So we can say there was a submerged condition of the sand. So they didn't get the uh, perfect amount of oxygen. So that's why the oxygen partial pressure fell to 0, and most of the eggs were dead when hatching occurred after 10 days later. So talking about the black-tailed prairie dog, so we all know that convective transport of gases, or we can say the bulk flow of the gases. So besides diffusion, the second major mechanism of respiratory gas transport is convection. So convective gas transport, also called the transport by bulk flow, occurs when a gas mixture or an aqueous solution flows and gas molecules in the gas or liquid are carried from place to place by the fluid flow. So the transport of oxygen by blood flow in an animal circulatory system can provide an example for that. So we all know that convection does not always require muscular effort from an animal. So we have got some ambient winds and water currents, in fact, that often move from oxygen and carbon dioxide from place to place in the environments of animals in ways that are very helpful. So as when global scale water currents renew the oxygen supplies of the deep sea. So sometimes these ambient winds and water currents even induce some internal flow within animals or animal-built structures. So explaining that in this induction of internal flow by ambient currents, in case of black-tailed prairie dog, so what happens is that animals are sometimes able to take advantage of these ambient currents or the winds or water currents in such ways that currents induce the fluid flow through their bodies or through structures they build. So prairie dogs, for example, they construct their burrow system in such a way that a wind that's flowing parallel to the ground surface is forced to rise and fall as it blows across some burrow openings, but not others. So because of the Bernoulli's principle, the pressure exerted at some burrow openings is lower than at others as a result. So there is a wind that's blowing in the parallel to the ground surface, thus induces the flow of air through the burrows, renewing this oxygen supply for underground. So for a more thorough discussion of this effect and other similar, we have to read about the cases of internal flow induced by the ambient current. So let's take an example of this, how we can explain the two types of these convective transport. So convective transport, which is also called the bulk flow. So there are two kinds of the precise effect of fluid flow on gas transport depending part of the geometry of the flow. So two geometries of importance are depicted in this figure. So one here you can see the unidirectional flow and the other is the tidal flow. So talking about the unidirectional flow, we can say that how the rate of convective gas transport can be calculated. Let's see as a model the simple case of oxygen transport by the unidirectional flow of a fluid through a tube. So you can see here in the figure A here. So the fluid passing through the tube, it explains the unidirectional flow. So when there is no exchange of gases across the walls. So this sort of model applies, for example, we can compare this to the oxygen transport by blood flow through the arteries of animals, because arteries basically act as conduits, neither adding oxygen to the blood passing through them nor removing oxygen. So we can compare this unidirectional flow in this tube to the blood flow through the arteries in the human body, in the animals. So in this case, where we can see that, so that we can calculate the rate of convective gas transport by CTF. So where CT represents the total concentration of gas in the fluid, and the that's chemically combined. For example, we have got oxygen combined with hemoglobin, and also the gas that's free in solution. And F here represents the rate of fluid flow. So talking about the tidal flow here, so in this case, you can see that there is a flow of this. For example, we can say that mammals and some other animals with very high demands for oxygen have favorable modifications of both these properties to enhance the rate of convective oxygen transport by the flow of blood through their arteries. So the second one is the this tidal flow. That means the back and forth flow can take place in and out of a blind-ended cavity. This we can relate with the lungs of mammals. So tidal flow is actually related to the lungs of mammals, and unidirectional flow takes place always when the blood is transported through the arteries. So what happens in this tidal flow is that we can say that back and forth flow happens. That means in and out of blind-ended cavity. So this is actually similar to the case in case of lungs. So moving on to the gas transport in animals, that often occurs via alternating convection and diffusion. So convection and diffusion are actually two kinds of the transport of gases. So convection is also called as the bulk flow. So gas transport in animals often occurs by both alternating convection and the diffusion. So convection, which is also called the bulk flow, and diffusion alternate in transporting oxygen from the atmosphere to the mitochondrion in a person. So how this happens actually, you can see, number first is the first step is the convective transport by flow of inhaled air from the atmosphere to the depth of lungs here. So the first step here, so the gas is inhaled, the oxygen gas is inhaled through the process of convection, and this is supplied to the lungs. So in second, you can see here the second step is the diffusion. That means diffusion occurs. So a gas-filled alveolar and sac, including the alveolus, then through the alveolar epithelium and the epithelium of a pulmonary blood capillary, and finally into a red blood cell where oxygen combines with hemoglobin. So only the final 0.00006 meters of this step requires diffusion through tissue or liquid. So initial some meters is covered by diffusion through gases. So that was the second step. So in the third step, there again, the third step occurs by convective transport by the circulation of blood. And last and the fourth step is that diffusion from an RBC, that's here. So diffusion from these RBCs in a systematic blood capillary to a mitochondrion in a muscle cell. So mitochondrion are actually much smaller relative to the muscle cell that has been shown. So this here, the system, in case of the humans, this explains how both the convection and diffusion transport takes place simultaneously. So there is alternating convection and diffusion. So you might have heard about the oxygen cascade. We can see the concept of the oxygen cascade is an especially informative way to summarize the transport of oxygen from the environment to the mitochondrion of an animal. So in everyday language, a cascade is a series of like steep waterfalls. So you can see here, so this is a cascading stream. So we can say that it resembles a waterfall that has different heights. So a cascade can be related to a series of steep waterfalls along a stream. Suppose that a water wheel or turbine is positioned. So there is this turbine or water wheel that's positioned at the lower side of this cascade. As you can see in the figure A, the force available to turn the water wheel is that available when the water drops from the height 4. So when the water drops from height 4 to height 5, there will be a kind of force that will move this turbine. So water starts at height A as it enters the cascade, but gradually as it flows through the cascade, it drops from 1 to 2, and then from 2 to 3, and then from 3 to 4, and then from 4 to 5 here. So final fall of the water provides the actual force to turn this water wheel. To construct an oxygen cascade, one plots the oxygen partial pressure at successive steps along the path between an animal's environment source of oxygen and its mitochondrion. So this is the oxygen cascade in people. So that was a cascading stream that I was giving you an example that how it actually, when the water flows and how at the final step it moves this turbine that turns up the water wheel. So this is the similar case in case of oxygen cascade in people. So because oxygen is not subjected to active transport, the partial pressure always drops. Here you can see the partial pressure of oxygen in atmosphere. So it's dropping from ambient air, then in alveolar gas, and then we have arterial blood, then average systemic, and then at mitochondrion, only the oxygen partial pressure that still prevails in the systematic capillaries is available to drive oxygen diffusion to the mitochondria. So why is the partial pressure that's plotted in an oxygen cascade uh, important? So the answer to this question is that essential concept for having a full understanding of physiology of oxygen transport. So mitochondrion of an animal are where the oxygen is ultimately used, as we have already seen in the last figure here. So mitochondrion is the final step where this oxygen is actually used. So oxygen enters the mitochondrion by diffusion. We already talked about that at a rate that depends on the difference in oxygen partial pressures between the blood in the capillaries and the mitochondrion. So just as we took the example of water wheels, so just as a water wheel on a stream requires water of certain height immediately upstream to function, so mitochondrion in each cell require a certain partial pressure of oxygen at the cell surface if they are to receive oxygen fast enough by diffusion to meet their oxygen needs. So moving on to the processes that affect the partial pressure of oxygen and carbon dioxide within a portion of terrestrial or aquatic environment. So we have certain respiratory environments. When we consider the respiratory environments of animals in nature, for example, biotic processes, they are as important as physical effects in determining the concentration and partial pressure of oxygen and carbon dioxide. So the processes at work in a portion of terrestrial or aquatic environment, as you can see in this figure, the animals and plants. living the animals and plants living there exert strong influences on the local concentration and partial pressure of oxygen and carbon dioxide. So we can say that during the day with adequate sunlight, there is photosynthetic organisms that add oxygen to the air or water and extract the carbon dioxide. So we have got certain animals, bacteria, and fungi here that remove the oxygen and add carbon dioxide. And at night, the photosynthetic organisms do the same. So any portion of the environment, as the figure shows here, exchanges the oxygen and carbon dioxide with neighboring regions by way of diffusion and convection, sometimes the breezes or water currents. So diffusion always tends to equalize partial pressure across the boundaries, and convection usually does so by physical mixing. So any portion of environment is common to find that the resident organisms collectively raise or lower the oxygen partial pressures. So that was all about the uh, introduction to oxygen and carbon dioxide physiology. So do you have any questions from this chapter? Okay, so moving on to the next chapter that we'll be talking about. So in this chapter, we will be talking about the external respiration, that is the physiology of breathing. So we will take an example of a tuna fish, and we will open this discussion with the example of tuna fish, how the external respiration takes place. So throughout the animal kingdom, species that depend on vigorous endurance exercise for survival, such as tunas, they must be able to acquire oxygen rapidly and steadily. So although the water is not a particularly rich source of oxygen, tunas have gills and breathing processes that enable them to live as highly active predators. So this is an here you can see in this figure a southern bluefin tuna. We call it, the zoological name of that is Tunus mccoy. So tunas actually represent one of the pinnacles of water breathing. So if they lived on land where they could be readily observed, they would be classified metamorphically with wolves. So African hunting dogs and other strong mobile predators. So judging by the length of time spent in motion, tunas are actually more mobile than any other terrestrial predator. So using their aerobic red swimming muscles, they swim continuously day and night. So the speed of like 1 to 2 body lengths per second. So some species, they may cover more than 100 kilometers per day during migrations. So to meet the relentless oxygen demands for their vigorous lifestyle, they require a respiratory system that can take up oxygen rapidly from the sea, and a circulatory system that can deliver oxygen rapidly from the gills to tissues throughout the body. So tunas actually breathe with gills, as do most of the fishes. So gills are hardly average. However, instead tuna gills are exceptionally specialized for the oxygen uptake. So illustrating a general principle in the study of respiration, that a single type of breathing system may exhibit a wide range of evolutionary refinements. So most fish, including the rainbow trout, drive water across their gills by a pumping cycle that's powered by their buccal and opercular muscles. So some species are there that adopt in alternating between the mechanism of ventilating their gills and other mechanisms that's called the ram ventilation. So fundamental concepts of external respiration. So in all animals, the system that's used to exchange respiratory gases with the environment can be diagrammed here in this figure. You can see that a gas exchange membrane or respiratory exchange membrane, a thin layer of tissue consisting typically of one or two simple epithelia, separates the internal tissues of the animal from the environmental medium. So external respiration or breathing is the process by which oxygen is transported to the gas exchange membrane from the environmental medium, and by which the carbon dioxide is transported away from the membrane into the environmental medium. So what we mean by the ventilation is actually the bulk flow or convection of air and water to and from the gas exchange membrane during breathing. So not all animals employ this ventilation. In some breathing can occur by diffusion rather than the convection. So we have the rate of diffusion across a membrane increases in proportion to the area of the membrane. So according to the fundamental diffusion equation, the rate of diffusion across a membrane increases as the thickness of the membrane decreases. So these physical laws explain why an expensive thin gas exchange membrane is a great asset for animals such as tunas that must acquire oxygen at higher rates. So in organs that are specialized for external respiration, the gas exchange membrane is typically thrown into extensive patterns of invaginations, or we can say the evaginations, which greatly increases the membrane surface area. So let's take the example of two kinds of these. We have got the external gills, we have got the internal gills. So gills are respiratory structures that are evaginated from the body and surrounded by the environmental medium. So here you can see the lungs. They are the invaginated into the body and contain the environmental medium. So these are the external gills that are evaginated from the body and project directly into the environmental medium. So we have got some internal gills also that are evaginated from the body and project into a superficial body cavity through which the environmental medium is pumped. So the internal gills are actually enclosed within a superficial body cavity, whereas external placement permits ambient water currents to flow over the gills. So internal placement usually requires an animal to use the metabolic energy to ventilate them. So when we talk about the internal gills here, so since they are evaginated from the body and project into a superficial body cavity, internal placement has its advantages, but they require and they use some metabolic energy to ventilate them. So when the gills are internal, the enclosing structures physically protect them and may help canalize the flow of water across the gills in ways that enhance the efficiency or control of breathing. So moving on to the tidal gas exchange, how the oxygen transfer from the environmental medium to the blood in a tidally ventilated lung. So like here in this figure, you can see that because a tidally ventilated lung is never fully emptied, fresh medium mixes in the lung with stale medium. So numbers are oxygen partial pressure in arbitrary units. The blood arriving at the breathing organ is assigned a value of 0, whereas the atmosphere is arbitrarily assigned a value of 100. You can see here. So what actually happens is that oxygen partial pressure in the blood leaving a breathing organ can in many ways be considered the best single measure of breathing organs effectiveness. So we have already explained that from the oxygen cascade concept that we already talked about. So to explore the implication of various designs, let's start by considering this tidally ventilated breathing organ, such as the lungs of mammals. So tidally ventilated breathing organs are distinguished by the fact that the medium, that means the air in this case, next to the gas exchange membrane is never fully fresh. So explanation is that such breathing organs are never entirely emptied between the breaths. So consequently, we can say when an animal breathes in the fresh medium, inhaled mixes inside the breathing organ with stale medium that's left behind the previous breathing cycle. And because of this mixing, the oxygen partial pressure of medium next to the gas exchange membrane is lower, often much more lower than the partial pressure in the outside environment. So oxygen partial pressure in the blood leaving the breathing organ is lower. So because a partial pressure gradient must exist between the medium next to the gas exchange membrane and the blood for oxygen to diffuse in the medium. So we can say that ideally ventilated structures, the oxygen partial pressure of blood leaving the breathing organ is below the oxygen partial pressure of the exhaled medium. So moving on to the formation and the action of H1F1, that means the hypoxia response of these elements. So we have one is the hypoxia H1F1 and other is the H1F2. So they are actually closely related transcription factors which binds with the specific hypoxia response elements. So most animals are fundamentally aerobic. So when the oxygen level in an animal's tissue becomes usually low, the low oxygen level typically means that there is some trouble. So animals detect this low oxygen level and respond to them at two different scales. So one scale of response to low oxygen uh, is that of the whole body, the scale of organ system. So the second scale of response is intracellular in cells throughout the body. So the intracellular response, or if the rate at which the breathing and circulatory systems supply oxygen, it fails to keep the pace with the rate of mitochondrial oxygen utilization. So that can result in cellular hypoxia. So this here we will be talking about the hypoxia inducible factor 1, that's HIF1. That means the hypoxia induced factor 1. And then we have the hypoxia induced factor 2. So these are the mostly central players in the intracellular responses. So they are actually closely related to the transcription factors. That means which binds with the specific hypoxia response elements that we have in the nucleotides of DNA to activate that gene transcription. So it has been found that in the last 2 decades, there has been an explosion of knowledge regarding these transcription factors, which are implicated in many disease states as well as playing crucial roles in the ordinary lives of animals. So HIF1 means, which means the hypoxia inducible factor 1, is an ancient molecule identified in mostly in the invertebrates as well as in all groups of the vertebrates. So what actually happens here is that the basic manner in which the intracellular concentration of these transcription factors are regulated is shown in this figure using the HIF1, that means the hypoxia-induced factor 1. So a complete hypoxia-induced factor 1 molecule consists of the alpha and beta molecules here. You can see alpha and beta subunits, we can say. The beta subunits are present regardless of the cellular oxygen level. So these are the alpha and beta subunits. Moreover, the alpha subunits are constantly being not only synthesized, but they are also tagged for breakdown by the ubiquitin. So we can say that we have the proteasome system, but also tagged for breakdown by the this. So when the oxygen level in all cell decreases, breakdown of alpha subunits is inhibited. So here you can see that there's some breakdown of alpha subunits because of the low intracellular oxygen, which inhibits the breakdown of these alpha subunits. And this here shows the synthesis of alpha subunits, and here promoting the dimerization of beta subunits with alpha subunits to form this H1F. That means the hypoxia-induced factor 1. So the concentration of these alpha subunits rises and more HIF1 molecules are formed by the dimerization of these alpha and beta subunits. So the HIF1 molecules enter the nucleus where they bind with the specific DNA. You can see. So these subunits, the alpha and beta subunits, they bind themselves with the DNA strand, where they bind with specific DNA sequences, inducing this hypoxia response element, which in turn control the gene transcription. So some species of animals are well adapted to living in environments where the ambient oxygen level is prone to fall to low levels. For example, we have the shark fish that lives in places where it experiences periods of very low environmental oxygen. So in a recent genomic study, researchers have asked whether the H1 HIF signaling system undergoes acclimation when fish uh, that have been living in high oxygen water begin to experience the low oxygen. So one can reason that if a fish that has been living in high oxygen water is about to confront a period of low environmental oxygen, the fish might profit by upregulating its HIF1 signaling system. So recognizing that it will depend on hypoxia-induced factor 1 signaling system to help its tissues to cope up with the hypoxia that they may soon experience. So using genomic methods, we can say that these so using genomic methods, we can say that researchers measure the expression in the heart muscles of genes that code for like hypoxia-induced factor 1 and other compounds in the H1F HIF signaling system. So talking about the total area and thickness of the gas exchange membrane in the gills or lungs of vertebrates as functions of body size. So the thickness of the barrier between the blood and environmental medium also shows significant evolutionary trends in the major vertebrate groups. So in most fish, you can see the sheet of gill tissue between the blood and water is about 5 to 10 micrometers. So vertebrate lungs uniformly have a much reduced barrier between blood and air, as you can see here in the figure B. So some fish, uh, you can see that the skin also varies widely. It also plays a wide role in gas exchange site in vertebrates. So some fish and most reptiles have evolved skins that permit little oxygen or carbon dioxide exchange. So in contrast, the skin can be responsible for 25% or more of gas exchange in other fish, in certain snakes and turtles, and sometimes in many amphibians or in the lungless salamanders, which not only lack the lungs, but they have an epidermis that is vascularized. So most mammals and birds, they resemble humans in relying almost entirely on their lungs. So moving on to the breathing organs of amphibians, we will be talking about the frogs, how they actually breathe. That means the amphibians of all the vertebrate groups, they mix water and air breathe. to the greatest extent. So many move from an aquatic environment to a terrestrial one during their individual development, and many are like dual breathers as the adults. So we can say that gills of aquatic amphibian larvae, like tadpoles, they are of different origin and structure than the gills of adult fish. So they develop certain outgrowths of integument of pharyngeal region and project into the water from the body wall. So what happens is that the gill that are external, you can see here in all the young, these adults that are external in all young amphibian larvae, in salamander larvae they remain so, but in the larvae of frogs and toads, uh, an outgrowth of integumentum, the operculum, that is different from the bony operculum of fish, soon encloses the gills in a chamber that opens to the outside posteriorly. So ventilation of the gills enclosed in the opercular cavity is actually accomplished by buccal pumping. So gills of amphibians are generally lost uh, at metamorphosis, but external gills remain throughout life in certain aquatic salamanders, such as mud puppies. So mud puppies ventilate their gills by waving them back and forth in the water and increase the frequency of these movements in response to the decreased oxygen or increased temperature. So the lungs of many adult amphibians, they are simple, well-vascularized sacs. Their internal surface area is increased little, if at all, by folding, and in this respect, they are less well-developed than the lungs of lungfish. So inner walls of the lungs of frogs and toads are often more elaborate. They may be thrown into complex patterns of interconnected folds in multiple tiers, giving them a honeycombed appearance. You can see here in the figure B. So exhalation in a frog results in part from the elastic recoil of expanded lungs and may also be promoted by contraction of muscles in the walls of the lungs. So exhalation is described as having both passive and active transport. So let's take an example of a bullfrog here, that's Rana catasbiana. So the bullfrog provides a well-studied specific example of pulmonary breathing cycle. So a bullfrog fills its buccal cavity in preparation for inflation of its lungs. So before it empties into the lungs to do so with its glottis closed, it inhales air, which mostly comes to lie in a posterior depression of the buccal floor. That's the first step here. So the first step here is it inhales air, which mostly comes to lie in a posterior depression. And the second step is that means the glottis is open and pulmonary exhalant air passes in a coherent stream across the dorsal part of the buccopharyngeal cavity to exit through these nares. So third step here, you can see. So this is the third step here. So what happens is the third step, the fresh air, when the buccal floor is, the fresh air in the depression of the buccal floor is then driven into the lungs when the buccal floor is raised with these nares closed. So an important effect of buccopharyngeal pumping between breaths is that it washes residual pulmonary exhalant air out of the buccal cavity, so that when the next pulmonary ventilatory cycle begins, the buccal cavity is filled with a relatively fresh mixture. So these are the different three steps in the ventilatory cycle of an adult bullfrog. So talking about the development of external respiration in the bullfrog, that's the Rana catasbiana. So bullfrogs, we can continue them with an example and start their lives without lungs when they are living at 20 degrees centigrade as aquatic tadpoles. So their gills and skin each account for about half of their oxygen and carbon dioxide exchange, like here you can see in this figure. So in adult frogs, the lungs take up most oxygen. And in tadpoles, the gills and skin each account for about half of oxygen exchange. So that's the oxygen. The y-axis here represents the oxygen uptake, and y-axis here represents the carbon dioxide excretion. So they're shown in this figure are the percentages of oxygen uptake and carbon dioxide excretion that occur across the gills, lungs, and skin of bullfrogs as they develop from tadpoles to adults. So animals, they were studied at 20 degrees centigrade and had free access to well aerated water and air. So talking about the breathing by mammals, so mammals and birds possess the most elaborate lungs of all animals. So their lungs are independently evolved and built on very different principles. So extreme intricacy of mammalian lungs is shown in this figure. So lung system of an adult human consists of 23 levels of airway branching. So the trachea first branches to form two major airways. We have the primary bronchi, that's the which enter the two lungs. The primary bronchi. Each primary bronchus then branches and rebranches dendritically within the lung and gives rise to secondary and higher order bronchi of smaller and smaller diameter, and then to even smaller fine tubes, which are known as bronchioles, like you can see here in this figure. So you can see in this picture, in this figure, that lungs on the left shows only the airways, whereas the lung on the right shows the airways and blood vessels. So the larger sizes of the major blood vessels reflecting the high rates at which blood must flow through the lungs. So to visualize the airways, the lung passages were injected with a yellow plastic. Similarly, the arteries and veins were injected with red and blue plastic respectively. So after this plastic hardened, the tissue was removed, leaving just the plastic to mark the airways and blood vessels. So only the airways and vessels of relatively large diameter are presented with this technique, so that might be clear to you. The airway system and the arterial and venous system branch far more finely than seen. So we have the arteries and veins that tend to branch in parallel with the airways. So respiratory airways of the mammalian lungs, talking about that, we have certain kind of, at the outer limits of this branching tree of airways, 23 branches away from the trachea, the frenal bronchioles, and blindly in alveolar ducts and alveolar sacs. So walls of which are composed of numerous semispherical outpocketings. So each called an alveolar, and collectively they are termed as alveoli. So if we say the singular kind of this alveoli, we can call it as alveolus. So the structure of alveolus is so elaborate that describing it is a great challenge and estimate. Uh, we will undergo a revision in this. So according to the latest values, there are almost 500 million alveoli in the two lungs of a human adult. So they vary in size, about average about 0.25 millimeter in diameter. So the trachea and bronchi and all, but the last few branches of bronchioles in mammals' lungs are not much involved in gas exchange. So these are known as the conducting airways and are said to constitute the anatomical dead space of the lungs. So talking about the total lung volume that's employed in different ways in different sorts of breathing. So when the mammals breathe fully, that means when they inhale and when they do not come close to deflating their lungs fully when they are exhale. So thus a wide margin exists for increasing the use of total lung volume. So tidal volume is actually the volume of air inhaled and exhaled per breath. So let's take an example of resting young men. The volume of the lungs at the end of the inhalation is about 2,900 milliliters, whereas that at the end of exhalation, it's about 2,400 milliliters. So the maximum volume of air that an individual can expel beyond the resting respiratory level is termed as the resting expiratory reserve volume. So in healthy young men, it's about 1,200 milliliters of the 2,400 milliliters of air. So in healthy young men, it's about, so let's take an example here that 1,200 milliliter can be exhaled by the maximum expiratory effort, but 1,200 milliliter, that's termed as the residual volume, cannot be exhaled at all. So maximum volume of air that can be inhaled beyond the resting inspiratory level is the resting inspiratory reserve volume. So in healthy young men, it's about 3,100 milliliters. Thus the total lung volume at the end of maximum inspiratory effort is about 6,000. You can see here in this picture. So volume attained by maximum inspiratory effort is like 6,000 milliliters. So using the terminology that we developed here, when mammals increase their tidal volume above their resting level, they do so by using parts of their inspiratory and expiratory reserve volumes. The maximum possible tidal volume, sometimes termed as a vital capacity, which is about 4,800 milliliters for this example. So talking about the mechanism of gas transport in the final branches of mammalian lungs during the inhalation. So we all know that gases in the alveoli, which is the gas that undergoes exchange within the blood, differs dramatically in composition from atmospheric air in all mammals. So we have the most fundamental reason is that the alveolar sacs from the blind ends of tidally ventilated airways are fully emptied. So the exact partial pressure of gases that prevail in the alveoli depend dynamically on the rate at which the fresh air is brought into the depth of the lungs and the rate of gas exchange within the blood. So ventilatory control system ordinarily adjusts the rate of ventilation relative to the rate of gas exchange with the blood. So the gas occupying the final respiratory airways of a mammal is essentially motionless. So this unexpected property is still another consequence of anatomical fact that alveolar sacs are the blind ends of a tidally ventilated airway system. So during inhalation, convective air movement carries air rapidly down the trachea and through the various bronchi. As the air flows ever deeper into the lungs, it slows because the collective volume of the airways rapidly increases as the airways branch into greater numbers here. You can see, so it's branching. So when it flows through the convection, the bulk flow is occurring. So it rapidly increases as the airways branch into the greater and greater numbers. So convective airflow ceases before the air reaches the alveolar sacs. So because the gas in the blind ends of airways is motionless, so oxygen and carbon dioxide transport through the gas must occur by diffusion. So talking about the airflow in the lungs and air sacs of the birds. So breathing by birds means the lungs of birds, although they are logical derivatives of the types of lungs thought to exist in the common ancestors of birds and mammals, they differ in their fundamental structural features from the lungs of mammals and all other modern vertebrates, except certain crocodilian reptiles. So structural difference between avian and mammalian lungs invites the comparison. So birds' trachea actually bifurcates to give rise to two primary bronchi which enter the lungs. So here the similarity to mammalian ants. So the primary bronchus that enters each lung passes through the lung, being known as mesobronchus within the lung. So two groups of branching means the secondary bronchi arise from the mesobronchus. So one group which arises at the anterior end of mesobronchus spreads over the ventral surface of the lung. So there is one more group that originates towards the posterior end of mesobronchus and spreads over the dorsolateral lung surface. So we call all these anterior and posterior groups of secondary bronchi. They are formally termed as medioventral and mediodorsal groups. So for simplicity, each group is represented as a single passageway in this figure. So we have the anterior and posterior secondary bronchi that are connected by a great variety of small tubes also. So let's take an example of air sacs of a goose and their connections to the lungs. So bird's air sac, which are part of the breathing system, are located outside the lungs, and they occupy a considerable portion of the thoracic and the abdominal body cavities. So usually there are about 9 air sacs that are divisible into two groups here. We have the abdominal sac, we have the posterior thoracic sac, anterior thoracic sac that connect to various anterior secondary branch bronchi. So posterior air sacs here, the posterior thoracic sacs, they connect to the posterior portion of mesobronchi. The air sacs are actually the thin-walled, poorly vascularized structures that play a vital role in gas exchange between the air and blood. So they are essential for breathing. So we have the structures of lung that describe are present in all birds, and their connections with the air sacs are similar. These lung structures, they are collectively called as paleopulmonal system, or simply we can say paleopulmo. So most birds in addition have a more or less extensively developed system of respiratory bronchial tubes. These are termed as neopulmonal system. So running directly between the posterior and the air sacs and the posterior parts of the mesobronchi and posterior secondary bronchi. So neopulmonal system is especially well developed in songbirds. The paleopulmonal system is always dominant. So we have a diversity of gills in aquatic, these invertebrates. So how do the breathing take place in aquatic invertebrates and other groups? So many small aquatic invertebrates and some large ones have no specialized breathing organs. They actually exchange gases across general body surfaces, which sometimes are ventilated by swimming motions or by cilia or flagella, generally warm water currents. So many larvae and some adults, they also lack a circulatory system. So thus gases, they move within their bodies by diffusion or by squishing of the body fluids from place to place. So we can say that these sorts of gas exchange systems are confined. So let's take an example of adults of the relatively advanced phyla of aquatic invertebrates typically have gills of some sort. So gills of the various major phyletic groups are often independently evolved, whereas they are all evaginated and projected into the water. They vary widely in their structure and in how they are ventilated. So mollusks, they actually exemplify an exceptional diversity of breathing organs. The phylum Mollusca nicely illustrates that within a phyletic group, a single basic sort of breathing can undergo wide diversification. So in mollusks, outfolding of dorsal body wall actually the outer body, in unfolding of the dorsal body will produce a sheet of tissue that's called a mantle, and that overhangs or surrounds all or part of the rest of the body, thereby enclosing an external body cavity that's called the mantle cavity. So one major modification of the gills in mollusks is the evolution of extensive sheet-like gills in the clams, or we can say oysters, and other lamellibranch, that's the sheet gill groups. So in these groups, 4 gill sheets are composed of fused or semi-fused filaments, like here you can see in the figure B. Cilia on the gill sheets dive incoming water through pores on the gill surface into water channels that run within the gill sheets. So in the cephalopod mollusks, the squids, cuttlefish, and octopus, it's not so much that gills that are specialized, but the mechanism of ventilation is very good. So gills are feathery structures. You can see here in this figure C, the gills or actually uh, these feathery structures that follow the unusual molluscal plan of being positioned in the mantle. So they are ventilated, however, the molluscal contraction rather than beating of cilia. So a final specialization worthy of note in mollusks is the evolution of the mantle cavity into the lung in the dominant group of snails. So these snails are actually called the group known, they are mostly known as the pulmonates. So in the terrestrial pulmonates, gills have disappeared and the walls of the mantle cavity have become highly vascularized and well suited for gas exchange. So some species are thought to employ the mantle cavity as diffusion line, but others ventilate by raising and lowering the floor at the cavity. So we have here a diversity of gills in certain aquatic invertebrates. So figure A, you can see here, is this is the teddy-bellied worm, a type of marine annelid that lives inside a tube. It constructs and can pump water in and out of the tube. So the figure B here shows the fan worm, another type of marine annelid who lives in a tube, but when undisturbed, it projects its well-developed area of pinnately divided tentacles into the ambient water. So these tentacles are used for both feeding and respiratory gas exchange. So they are ventilated by the action of cilia on the tentacles. So number C here is in this figure is the C star, that's with branchial uh, papillae and tube peat that are used as gills. So sea stars, they bear many thin-walled finger-like projections from their cavity, termed bronchial papillae, on their upper body surfaces. So respiratory gases pass between this fluid and ambient water by diffusion through the walls of the papillae. Last is here the horseshoe crab that shows a single gill that's composed of many gill sheets. So horseshoe crabs, those are also called as limulus. They have unique book gills consisting of many thin gill sheets arranged like pages of a book. So the book gills are protected under thick gill plates, which undergo rhythmic flapping motions and that ventilate those gills. So that was all about the second chapter. That's the physiology of breathing. So moving on to our third chapter, which is the transport of oxygen and the carbon dioxide in body fluids. So we will be talking about how the transport of oxygen and carbon dioxide in body fluids that take place. So starting with an example of a human fetus. which obtains oxygen by pumping blood along its umbilical cord to the placenta, where its blood picks up the oxygen from its mother's blood. So hemoglobin here plays a major role in the acquisition and transport of oxygen by the fetus. So in the mother, the oxygen taken up in her lungs combines with hemoglobin in her red blood cells and is carried by blood flow to the placenta in that form. So in the placenta, the fetus's hemoglobin combines with oxygen that is released from the mother's hemoglobin. So the oxygen that's combined with the fetal hemoglobin is then carried out by the circulation of the fetus's blood from the placenta into the body of fetus, where the oxygen is used. So how this happens is that a developing mammalian fetus, it receives oxygen and voids carbon dioxide by means of a close juxtaposition of its blood vessels within those of its mother's circulatory system in the placenta, as the fetus is always attached to the placenta of her mother. So the fetus's heart, it pumps the blood through the umbilical cord, which is this one here, the umbilical cord, through which the fetus gets everything from his or her mother. So So fetus's heart pumps blood through the umbilical cord to the placenta, where that blood picks up oxygen. That's its mother's blood has brought there from her lungs. So now this oxygenated fetal blood returns to the fetus through the umbilical cord and is circulated to all parts of the body. This is how the fetus gets the oxygen and other food and other things from her mother. So by 10 weeks after conception, a human fetus already has the hemoglobin-rich blood. So at that age, amount of hemoglobin per unit of volume in the fetus has reached like 50% of the adult concentration. And it's increasing rapidly. So that it will be about 80% of the adult value at 20 weeks of age. So what happens in the placenta, the oxygen must cross from the mother's blood to the fetus's blood by diffusion through tissues that are separating the two circulatory systems. That means one is of the fetus and other the mother. So the detailed way in which this occurs remains a topic of active research. So basic options for the mode of gas transfer or the gas exchange or cross current gas exchange and concurrent gas exchanges. So the mode of gas transfer in the human placenta uh, remains uncertain for some of the reasons. First, the experiments cannot be done on human fetuses. Second reason might be that other species of placental mammals exhibit such wide diversity in the morphology and physiology of their placentas that researchers are not certain which animal model would best reveal how the human placenta works. So the hemoglobin are one of the several types of respiratory pigments or oxygen transport pigments that animals have evolved. So we have got the two hemoglobin or other respiratory pigments that combine with oxygen. They are said to be oxygenated, and then when they release oxygen, they are deoxygenated. So they are not said to be oxidized and reduced, but we can say that the hemoglobin that's oxygenated, and when they release that oxygen, they are called deoxygenated. So how the absorption spectra of respiratory pigments takes place. So to measure this absorption, light of each wavelength is passed through a hemoglobin solution of definite concentration, and optical path lengths in this case here are shown. The concentration was like 1 millimolar, uh, and the light path through the solution was 1 centimeter. The fraction of the incoming photon energy that fails to pass through the solution is measured. So from this data, one can calculate the extinction coefficient, which is a measure of absorption of light by hemoglobin. So talking about the chemical structure of hemoglobin, hemoglobin contains heme and are the most widespread respiratory pigments. So chemical structure of all hemoglobin molecules They share two features. First, all hemoglobins contain heme, that means Heme, which is a particular metalloporphyrin containing iron in the ferrous sulfate. Second is non-covalently bonded protein that's known as globin. So that makes it hemoglobin. Like figure here, you can see this is the hem and figure B here is the globin, and when they combine together, this forms the hemoglobin. The combination of heme with globin accounts for the name hemoglobin. Oxygen binds at the heme site at a ratio of 1 oxygen per heme. So in all hemoglobin molecules, the heme is identical. So the blood hemoglobins of vertebrates are almost always 4-unit molecules. Like here you can see in the picture. So it has two alpha globins and these two beta globins that can bind a total of 4 oxygen molecules. So two types of globins termed as alpha globins and the beta globins here are found in adult hemoglobin. So each molecule of adult hemoglobin consists of 2 alpha units and 2 beta units. So the human alpha globin contains about 141 amino acids, whereas the human beta globin contains about 146 amino acids. So all the other vertebrate species are also described having both these alpha and beta globulins. The particular chemical structure vary from species to species. So relatively, we can say that huge hemoglobin molecules are found in the blood of some vertebrates. So here comes the distribution of the two major respiratory pigments. We have the hemoglobins and hemocyanins. So a red square here indicates that hemoglobin occurs. You can see the red squares here. So these actually indicate that hemoglobins occur in solid tissues such as muscle or nerve tissues. So red circles, you can see some of the red circles here. These indicate the hemoglobins occur in circulating body fluids. So a single small red circle indicates the presence in circulatory blood fluids of hemoglobins, consisting of 1 unit molecule of heme plus globin. So moving on to the next slide, we will talk about the typical oxygen equilibrium curve for human arterial blood presented in two different ways. So a key tool for understanding the function of a respiratory pigment is the oxygen equilibrium curve. So in a body fluid containing the respiratory pigment, there is a large population of oxygen binding sites. So human blood, for example, it contains about 5.4 into 10 raised power 20 hemigroups per 100 ml. So combination of these oxygen with each individual oxygen binding site is stoichiometric. One and only one oxygen molecule can bind, for example, with each hemigroup of hemoglobin or with each pair of copper atoms that are present in the hemocyanin. So however, in bloods, there are a great number of oxygen binding sites. All sites do not simultaneously bind with oxygen or release the oxygen. Instead, we have the fraction of oxygen binding sites that are oxygenated with the oxygen partial pressure. So oxygen equilibrium curve, also sometimes called as oxygen dissociation curve, it shows the functional relation between the percentage of uh, the binding sites that are oxygenated. So x-axis here, the percentage of hemigroups that are oxygenated, or we can say the percentage of binding sites that are oxygenated, and also the partial pressure of the oxygen in the blood. So respiratory pigment in the blood of an animal is said to be saturated if the oxygen partial pressure is high enough for all oxygen binding sites. So the oxygen equilibrium curve can be presented in two ways, showing the percentage of oxygenated binding sites as a function of oxygen partial pressure, The alternative presentation is seen in this figure here. So this is the oxygen equilibrium curve that shows the total amount of oxygen per unit of blood volume, including both the hemoglobin bound and dissolved oxygen. And here it reaches a plateau that is a flat part here is the oxygen equilibrium curve. That means a wide range of blood oxygen partial pressure is sufficient to cause full hemoglobin saturation. So here you can see the x-axis, it represents the partial pressure of oxygen in blood, and the y-axis represents the milliliter of oxygen per 100 ml of blood. So talking about the oxygen delivery by human blood at rest and during the vigorous exercise here. So the oxygen equilibrium curve shown is here is that for human arterial blood, like here in this figure, you can see the thick shaded areas on the curve show representative ranges of blood oxygen concentration and oxygen partial pressure in the lungs. That's the blue one. So the systemic tissues during rest, that's the green one, and the systemic tissues during vigorous exercises, that's the vertical purple area here. Arrows to the right show how much oxygen is delivered to the tissues by each 100 ml of blood during rest and exercise. So all values are actually the semi-quantitative intent of the diagrams. So effects of pH and other variables of blood hemoglobin, they are not included in this. So arrows here show the drop in blood oxygen concentration as the blood moves from the lungs flow through the systematic tissues. So this shaded portion here, it shows the oxygen in ml that's released to tissues by each 100 ml of blood. So let's talk about the Bohr effect. So oxygen affinity depends on the partial pressure of carbon dioxide and the pH. So in a body fluid or tissue containing a respiratory pigment, there is a decrease in pH or an increase in carbon dioxide partial pressures. So that often causes the oxygen affinity to decrease, thus shifting this oxygen equilibrium curve to the right. So this effect is illustrated in this figure here. So the affinity for oxygen decreases as the blood pH decreases. As affinity of decreases the oxygen equilibrium curve, it shifts to the right. So this is called the Bohr effect, which was named after the scientist Christian Bohr in 1855 to 1911. So this Bohr effect is actually results from the influence of proton concentration or respiratory pigment molecules, and a carbon dioxide Bohr effect results from the immediate influence of increased carbon dioxide partial pressure. So moving on to the next slide, we will talk about the root effect in eels. So how that happens is that acidification sometimes lowers the oxygen carrying capacity of hemoglobin. So you can see here that in some types of animals, because of the distinctive properties of their respiratory pigments and increase in carbon dioxide partial pressure or decrease in pH of blood, not only causes a Bohr effect, but also reduces the amount of uh, oxygen in the respiratory pigment that binds when they're saturated. So reduction in the amount of this oxygen uh, bound to the pigment at saturation is termed as the root effect, which was named after a scientist whose name was Root. So root effect provide a mechanism by which the oxygen partial pressure of even well-oxygenated blood can be dramatically increased under the control of blood pH. To see this, consider the hemoglobin in the blood of eels. So when it is fully loaded with oxygen, here in this figure you can see at a pH of 7.54, the hemoglobin is chemically combined with about 12.6 ml of oxygen per 100 ml of blood. So acidification to a pH of 7.3, it lowers the oxygen binding capacity of the hemoglobin because of the root effect, so that the hemoglobin can chemically combine with only about 9.4 ml oxygen per 100 ml. So in this way, we can say that oxygen is forced off the hemoglobin by the change of this pH. So pH is said to have pH of the blood is said to have a pronounced effect on this root effect. So acidification from pH 7.54 to 7.35 forces the eel hemoglobin to unload 3.2 ml of oxygen into each 100 ml of blood. So this oxygen released goes into the blood solution. So it has no other immediate place to go. So by dissolving, it dramatically elevates the blood oxygen partial pressure. So this is what we call as the root effect. And we talked about this root effect in case of the eels. So talking about the organic modulators that often exert chronic effects on oxygen affinity. So we will talk about the resurrection of blood hemoglobin of extinct woolly mammoth. So for ancient adaptation to the challenges of regional hyperthermia, how they adopt to this uh, climatic conditions. So using the genomic methods, researchers have recently resurrected the hemoglobin of extinct woolly mammoth. So they could study the hemoglobin directly. So mammoth was an abundant resident of Arctic and subarctic environments. So in sharp contrast to its extant relatives, the African and Asian elephants, Arctic mammals often permit tissue temperatures in their appendages. So raising the possibility that oxygen affinity of hemoglobin might be raised to such a high level by low tissue temperatures that the appendage tissues are subjected to impaired oxygen offloading. So was that a problem for woolly mammoths? So by studying the resurrected mammoth, hemoglobin researchers concluded that in fact the uh, blood hemoglobin of woolly mammoth had adaptive speciations that made it relatively insensitive to these low temperatures. The researchers, they extracted actually the DNA from a 43,000-year-old permafrost preserved mammoth that was collected in Siberia. They then amplified and sequenced those genes in the DNA that coded for the alpha and beta globin chains of hemoglobin. So the researchers then introduced the genes for Asian elephant globins into the bacterium Escherichia coli, which faithfully synthesized these elephant hemoglobin. Finally, by the use of site-directed mutagenesis, the researchers modified the introduced elephant genes at the positions that need to be changed for the E. coli synthesized. So the globin proteins to match these once circulated in the blood of extinct woolly mammoth. So thereafter, the E. coli that produced the authentic woolly mammoth hemoglobin, which was purified and studied to determine its thermal and oxygen transport properties. So when water fleas are transported to oxygen or transferred to oxygen poor environment, their oxygen transport system undergoes rapid acclimation because of altered gene expression. So how actually that happens is that the water fleas, that's the Daphnia magna, had been living in a valley of water and were transferred at times uh, at time zero to water in which the oxygen partial pressure was only 15% as high. The composition and concentration of their blood hemoglobin were then monitored for 11 days. So the animals change color as shown by the photographs here. You can see, so these animals, they are actually changing the colors. So talking about the blood and circulation in mammals at high altitudes, So study of blood oxygen transport in humans and other mammals at high altitude in its particular way, one of the most intriguing chapters in the evolutionary physiology. So this is true because in the past 30 years, the blood responses of lowland people at high altitude have morphed from being touted as exceptional examples. So if you think back to the oxygen cascade that we talked about for people in high index, you will recall that when people, whether native lowlanders or highlanders, are exposed to the reduced atmospheric partial pressure of oxygen at high altitude, they actually do not experience an equal reduction in their venous oxygen partial pressure. So venous partial pressure in fact is reduced far less than the atmospheric pressure. So that might be the key reason for this conservation of oxygen partial pressure in blood oxygen transport. So the drop in oxygen partial pressure is actually between these arterial and venous blood is smaller at high altitude than at the low altitudes. So talking about the major processes of carbon uptake by the blood, here you can see that the molecules that are boxed in red and purple represent the oxygenated and deoxygenated hemoglobin. So three elements of molecular structure are highlighted in each hemoglobin molecule. The oxygen bind siting is the HB, symbolized as HB. So we have some buffering groups that can participate in carbamate formation. So the process shown in this figure occur in reverse in the lungs or gills, whereas the labels refer to the site of buffering. So they are specifying where the H positive generated during bicarbonate reduction is taken up by the buffering compounds. So carbamate formation actually occurs to a significant extent in mammals, but not necessarily in other invertebrates. So rising atmospheric carbon dioxide acidifies the oceans, and how this happens is that in the study of global climatic change, which is a concern that has recently become a primary focus in ocean acidification. So acidification caused by carbon dioxide is a more recently identified challenge. So carbon dioxide concentration of Earth's atmosphere has risen from about 300 parts per million to about 400 parts per million in the past century because of the burning of wood, coal, petroleum. So because of these principles, we have discussed that carbon dioxide concentration in the waters of the ocean has increased. This has made the oceans more acidic by about 0.1 pH unit by driving the reactions that were shown here. So animals are not always able to regulate processes that are affected by the environmental acidification. For example, acidification tends to erode certain types of animal skeletons that are composed of calcium carbonate. For example, we have the skeletons of reef corals, or to interfere with production of such skeletons. It can also interfere with the sensory and developmental processes. So that was all about the acidification of aquatic habitats, how it's leading to the global climate change. Okay, thank you so much. That was all about this week's lectures.